Hi, my name is Maria Trifas, and I'm a senior in Columbia College studying biochemistry. This summer, I worked with the Henssen group at Charité Hospital in Berlin, Germany, and my project focused on understanding the role of ecDNA-mediated genes in a neuroblastoma cell line. So I looked at ecDNA, which is a very interesting form of DNA that's found in human cancer cells, and it's essentially weird because it's circular, whereas most human DNA is linear, and it also often contains many oncogenes or genes that are known to drive the aberrant growth of cancer. So my main research questions with my project were, are differences in this ecDNA amplicon counts reflected in overall transcriptional profiles of cells, and what cellular pathways are affected by the increased ecDNA expression within tumors. So we sought to answer these questions through single-cell RNA sequencing, which provides a snapshot of the transcriptional profile, like which genes are being expressed in a cell. So I focused my analysis on single-cell RNA sequencing data from a CHIP212 neuroblastoma cell line. And I first clustered the cells both by cell cycle expression and by MYCN expression. And interestingly, we found that their MYCN differences weren't the main source of heterogeneity between the cells in the sample, but all of the cells with MYCN amplification that we found and classified as MYCN high cells also amplified the other genetic targets on the ecDNA MYCN amplicon. So from this result, we saw that the increases in MYCN expression that we saw were likely due to increased presence of ecDNA. So we further ran pathway enrichment analysis to see what specific pathways differed between these MYCN high expressing cells and MYCN low expressing cells, and we found that the MYCN high cells were more likely to be dividing and in mitotic parts of the cell cycle, and also had high MYCN target gene expression, whereas the MYCN low cells had a more developmental phenotype with the expression of extracellular matrix factors, tumor suppressors, and even developmental genes. So this analysis pointed to some of the differences as a result of varied ecDNA expression, and it sought to inform how these differences really cause different changes and heterogeneity that we see within tumor